So, today we're diving into one of the most incredible team-ups in science right now, the fusion of artificial intelligence and gene editing. We're going to explore how this combination isn't just making science faster, it's completely changing what we thought was even possible.
Let's just start with a huge question, one that honestly felt like pure science fiction until just a few years ago. What if we could actually go into our own DNA and fix the tiny errors that cause devastating diseases? Well, guess what? That's not sci-fi anymore. It's happening in labs all over the world as we speak.
And the technology at the heart of all this? It's called CRISPR. You know, the easiest way to get your head around it is to think of the find and replace feature in a document. Except here, we're talking about our genetic code. It lets scientists find one specific stretch of DNA and then cut it, swap it, or fix it with amazing precision. It's basically a pair of molecular scissors.
Look, there's no doubt that when CRISPR first came on the scene, it was a total game changer. It held out this incredible promise of curing genetic diseases, creating stronger crops, you name it. But even with all that potential, there was a pretty big roadblock standing in the way.
So here's the thing. Not all genetic fixes are the same. For different jobs, scientists needed different kinds of CRISPR tools. Some needed to be big, some small, some needed to make a cut, others just a tiny tweak. But actually finding these different tools out in the wild of nature, it was like looking for a needle in a haystack, a really, really slow and difficult process done by hand.
And this is where the story gets really wild. Enter AI. Instead of people having to manually search through just mountains of genetic data, researchers thought, hey, what if we train an AI to do the hunting for us? And the results, they were absolutely staggering.
Just look at the difference here. It's night and day. Researchers over at MIT gave an AI this exact task, a job that would take a whole team of scientists weeks, maybe even months, just to find one new tool. The AI did it in minutes. And it didn't find just one, it found hundreds. Think about that for a second. More than 200 new gene editing tools, all discovered in a single pass. This wasn't just a small step forward, it was a giant leap. All of a sudden, the toolkit scientists had went from, a hammer and a screwdriver to a massive, high-tech workshop filled with specialized gear.
But here's the thing. It wasn't just about finding more tools. It was about finding the right ones. Tools that had the specific features needed to solve some of the biggest challenges in gene therapy. And one of the most stubborn of those challenges was just plain old size.
You see, to fix a genetic disease inside a person, you have to get that CRISPR editor into their cells. Scientists often use harmless viruses to make the delivery, but most CRISPR systems were just too big to fit inside. It was literally like trying to stuff a grand piano into a shoebox. It just wasn't going to work.
And this is why that AI discovery was so huge, because hiding in that treasure trove of hundreds of new tools was this family of miniature editors called Cas12F. They're super compact, tiny enough to actually fit inside those delivery vehicles. This was the key that could finally unlock treatments inside the human body.
So let's walk through how this actually works. With these tiny tools, the whole process is now possible. First, you package the mini CRISPR system into a safe delivery vehicle. Then, you introduce it to the patient, and it travels right to the target cells, maybe in the liver or the eye, and it edits the faulty gene right there, at the source of the problem. This is a complete game changer. Okay, so this tech can be used for tons of things, but let's zoom in on one really urgent global crisis where it's already making a difference: the fight against antibiotic-resistant superbugs. This is a huge deal. We're talking about more than 700,000 deaths every single year because common infections have learned to fight off our best drugs. It's like a silent pandemic, and it threatens to throw modern medicine back into the dark ages. But CRISPR, it offers a totally new way to fight back. And here's the incredible part. Scientists have actually used CRISPR to reverse that resistance in bacteria. Essentially, they figured out how to use this gene editing tool to disarm the superbugs, making them, well, not so super anymore.
So how do they do it? Well, they program the CRISPR system to hunt down the exact genes that give the bacteria their superpowers, and they just snip them out. By deleting that piece of code, the bacteria become totally vulnerable to our old antibiotics again. We don't even need new drugs. We can just make the ones we already have powerful again.
This is all just amazing, right? This team-up of AI and CRISPR is opening up a whole new frontier in biology. But like with any technology this powerful, we have to talk about the other side of the coin, the new risks and responsibilities that come along with it.
So we've seen how AI can be this incredible force for good, but you have to ask yourself, what happens when that same power is pointed in the wrong direction? When the lab itself can run on its own, what kind of new dangers pop up?
And recent reports are highlighting some pretty serious biosecurity risks. For one thing, these same AI systems could potentially be used to help design dangerous new pathogens. Then you combine that with automated labs, where experiments could be run without anyone even there, and the risks really start to stack up. And maybe the scariest part? This power could become available to people with very little training in biology, which really lowers the bar for someone to misuse it.
There is just no question about it. AI and CRISPR are fundamentally rewriting the code of life, and along with it, our entire future. We are getting this unbelievable power to cure diseases and fight plagues. But in the process, we're also creating tools that carry unprecedented risks. The science is moving at lightning speed, and that leaves all of us with one final and really important question. Are we, as a society, actually ready for what's coming next?